Hello everyone, thank you for joining us and welcome to Horizon's presentation webinar jointly with Thirion around partnering to offer high efficiency shRNAs and overexpression reagents. First I'd like to introduce the speaker today, Dr. Christian Thirion. Dr. Thirion is one of the founders of Thirion Biotech, and he'll be just spending quite a bit of time talking about their unique product offering and service offering in the area of shRNAs. And with that, I will pass presentation over to Dr. Thirion.
So, thank you, Chris. So I hope everyone can see, okay, my screen now. Okay, so I'm really glad to present now our technology and also not to mention our collaboration that Thirion and Horizon started. So our intention was to combine at least Thirion's technology with Horizon's outstanding genome editing technology. It's like if you have a target research question and you would like to have a clear answer, is the target really associated with the phenotype? So this will it be about today in the presentation, and I will start right on just to give you what's on the menu. So of course not everyone knows Thirion Biotech, so we are a European biotech company focused on viral vectors technology. So I will shortly introduce the viral vectors technology that we use, basic technology and applications, and then I will follow on with our core competence, which is the cell modeling using lentiviral vector technology. And here you will see that the RNAI-AND platform is really fitting exactly in the target validation stream. So imagine you start with an siRNA screening, you have some hits, you want to confirm that the hits are really good so that the targets really are associated with a phenotype. Then RNAI-AND comes into play. So we will generate knockdowns which are of the highest efficiency and then use lentiviral technology to provide cell models which have built-in controls. And this will be the final slide deck about VARICHEK, our target validation cell model. All these are available at Horizon, and as you are all aware, this is an intermediate step, and then you can follow on with gene editing, and ultimately you have access to a very powerful screening cell model. Let's go. So not too many words on the company. The company has been founded in 2005, and I am one of the founders. So I come from the viral vector technology and field of adenoviruses. I worked a long time modifying surfaces and developing vaccine vectors. So it is not surprising that a part of Thirion's activity is also dedicated to viral vectors, especially human novel adenoviruses, which are rare for vaccine development. They will not be part of the presentation today. And one of my co-founders, so of course we are only two co-founders, so the co-founder, he came from the gene knockdown and cell biology. So we combined in 2005 both worlds, and we generated cell models which had a very high degree of knockdown and used viral vectors to transport the expression cassettes into those cells, and we can do it in vitro and in vivo. Okay, and since we are working with viral vectors, and viral vectors have a very high transduction efficiency for human primary cells are good targets and can be modeled using those tools. Okay, I'll try to move on with the presentation, which is a little bit stuck now. Okay, here we are. Okay, so the preclinical research challenge that we focus on is the target validation bottleneck that our pharma drug discovery has. So you have many hits, but the choice of the right target to develop your small compound is critical, and there is often failure. What we do is that we provide certainty at the level of in vitro models. I will present those two models today in detail. And the other aspects, like working in primary cells, keep it in mind that you can use viral vectors very nicely to work in primary cells, and through Horizon all the viral vectors are of course available. we have a predictive in vitro model which is emerging. This is 3D cell modeling, and we have access through collaborations and can also assist you in generating 3D cell models if wanted, but this is very early stage and few early adopters are using 3D in vitro models for screening nowadays. An emerging topic is drug resistance, so point mutations, and again, in order to just have an idea and confirm if the point mutation is really associated with a phenotype, one can also think about using the VARICHECK model, which has an inducible knockdown combined with an inducible expression of the protein mutant. You will see some data on this during the presentation. This is where we are located. It's a very complex value chain, so there are many players if you want to go from the target research to the first lead and then enter a preclinical study. We mainly serve the early target discovery, screening, and then some of the hit lead identification steps. And what we did is Horizon and Thirion teamed up, and we provide all the RNAi tools and lentiviral tools within the Horizon workflow of target research. So that's going to be a really good solution for everyone. Viral vector technology. Okay, what is it about? There are 3 viral vectors which are currently in use in, I would say, many, I would even say all the basic molecular laboratories working with primary cell cultures and associated with target research. Adenoviral vectors, they are very strong in generating a strong gene expression, and it's transient. This means the adenoviral vector will transduce 95% of the epithelial and endothelial cell lines quantitatively, strongly express the gene of interest, or in this case knockdown, in this case CDK5 in UVEX, and then you see that after a couple of days the knockdown signal simply comes back. So this is the knockdown signal at day 4, then it comes slowly back at day 6 and day 8. This means that the viral vector is episomal, it is diluted out when the cells proliferate. This has some beautiful advantages and can be used when, for example, primary cells cannot be stably selected. Lentiviral vectors are the ideal tool when you work with established tumor cell lines, and they generate high gene expression, even highly regulatable gene expression, and I will show you our lentiviral vectors that we developed and also homogeneous expression. The AAV vectors, of course, they are used in vitro. You can use them using the X-Men, for example, product line of Horizon for genome editing. But AAV vectors can also be used in vivo, and as you can see here, they can be rendered even tissue specific, and that is also a service that we offer. Just to sum up a little bit our USPs, so we have a kind of core facility at Munich, in Cologne, and another in Heidelberg, so it's Germany. We provide viral vectors which are high titer for preclinical research. All are very highly controlled, so the quality is very high. We can even supply qualified material meeting regulatory standards. We have a cell line which has a cGMP master cell bank. We have AAV vectors which we can highly purify for central nervous system applications, and we have developed own reagents for cell-specific gene delivery and enhancement. What is we worked really hardly in the past years to offer the highest degree of customization possible. This means that we can not only produce in very short time frames ranging from 4 to 5 weeks for the 3 vector types, but we can also accommodate TET-inducible gene expression and of course our knockdown technology and all the reporter gene technologies. Okay, so now we're going to start with a few slides on AAV vector and adenoviral vectors, and then we're going to really go into the technological details. Adenoviral vectors, they are non-pathogenic and have a very low immunogenicity in vivo. What is outstanding is their long gene expression up to 10 years by the site of injection. And it's a very small particle. You can even call it a nanoparticle with 25 nanometers in diameter. So it's really amazing. You can package 4.5 kb, and it is so good that it has even succeeded in a first gene therapy. Nowadays there are more than 80 clinical trials worldwide involving AAV vectors, and there's a good bet that there will be more approved AAV-based therapies than the one in 2012 approved for Unicure for lipase deficiency. A few things you need to know about AAVs. They come with different types, so the code is different, and there are 6 types that we produce, and each type has a certain tropism, which means that the vector preferentially goes into certain tissues when injected systemically. Liver, for example, is very good for AAV2, and the central nervous system, for example, we offer the standard AAV1-2 hybrid vectors. Skeletal muscle is very good also with AAV1, and AAV6, for example, is excellent for heart. So there are some flexibility regarding the target organ. This is one example. What does it look like in real life? This is a large animal model, porcine animal model, where we combined a tissue-specific promoter with AAV6, and here you see that descending the coronary artery, you have a beautiful GFP signal, which means that you have really nicely transduced the tissue or the organ of interest. And this stays for more than 1 year stable in gene expression. Couple of words to adenoviral vectors. So they are non-enveloped double-strand DNA viruses, and what we use an E1 and E3 deleted vector. It is immunogenic in vivo, therefore we prefer to use it in vitro, and we use the serotype 5 as a commonly used gene delivery vector. It can package up to 7.5 kb of foreign DNA. All the customization options that we offer are sufficient for any experiment you would plan. So there is knockdown, microRNA expression inhibition, there is inducible AAV vectors and adenoviral vectors, there is adenoviral vectors expressing all kinds of cDNAs or IPS. There is even new adenoviral serotypes that we offer. So there's really a large product opportunity and portfolio that Thirion offers. And coming to the reagent, so this is the reason why we became specialists. There were cells, for example, that were not transducible by lentiviruses or adenoviruses, and in this case we even developed reagents to make them transducible. So in this case the primary receptor is lacking, for example, in non-human cells like murine cells. Concentrate on the NIH3T3 example. So without our adjuvant you get a poor transduction. If you add the adjuvant you get a beautiful transduction and very high gene expression. This is because in NIH3T3 cells the primary attachment receptor is not working well, and therefore you need a bridge which binds to the vector on the one side and to the cell surface to reestablish this expression, and we get in general 20 to 50-fold enhanced gene expression when applying this reagent. Okay, lentiviral vectors. This is a complex chart. I will not go into every detail for every genetic element, but it's important for you to understand a little bit the biology behind it. We have lentiviral vectors are essentially free of lentivirus genes, because that's always a safety point and issue that we are asked, is it free of lentiviral or retroviral genes? So yes, the answer is yes, and it is completely inactivated since we use the latest generation. Here we have a deletion and delta U3 deletion, so all the intrinsic activity of this promoter is lost when the lentivirus is integrated in the genome. And then we have space for 8 kb foreign DNA, so this is the size of your expression cassette, which is then the only expression cassette that is transferred and integrated into their host genome. Okay, now as promised, I really start with details and science about SIRION'S vector technology. First, if you work with lentiviruses, you have always to make sure that the expression is stable and not lost. Because if you have like genes which are a little bit toxic or which cause the cell to get slowed down in cell growth, you have in heterogeneous cell populations, you always have kind of a very heterogeneous resulting stable cell pool. What we did, and this is standard with SIRION, we coupled the expression of the gene of interest in blue via an internal ribosomal entry site to a puromycin selection marker. So this cassette is really essential for the success and use of lentiviral vectors. The promoter is also important because we choose promoters which are constitutively active in all the cells, or in, let's say, in all the tumor cell lines, and not silenced. What you can now contemplate below is we have two cell lines where we express the receptor tyrosine kinase, and this is not a clonal cell line. This is a cell pool where we have simply added the lentivirus expressing this receptor tyrosine kinase and then selected with puromycin for 2 weeks. So it's a polyclonal cell pool. It's very fast. Within 4 weeks we have this cell product, and what you see here in the FACS profile, you see that this is the parental cell line without lentivirus, and then we added the lentivirus, and then we get a very nice, sharp, highly expressing cell population, but very homogeneous. And this is achieved when combining exactly these two elements, for example, using the iris. So those vectors, of course, we have a huge set of vectors ready to use, expressing validated shRNAs, which Horizon offers. Now we went one step further. We improved, starting from this lentiviral vectors, which are beautifully applicable for overexpression cell models. We improved now the system in order to generate a 3G TET-inducible cell system. So what the hell is 3G TET-inducible? So 3G stands for the third generation of TET-inducible promoters and transactivators. Okay, so it's the latest, it's very tight, and it's very highly inducible. We optimized the LTRs. We coupled, as I showed you, the gene of the expression of the gene of interest with a kind of resistance marker in order to maintain the expression of the gene. Then we have a choice of specific promoters to prevent silencing in cell lines, and we have a tight regulation with the latest 3G technology. Now I'll show you the result. This is our all-in-one inducible lentiviral vectors. The blue element is the classical SIRION expression cassette, where we couple, in this case, the transactivator with the antibiotic selection cassette. Here we have a first promoter, which is conditionally active in the cell line we want. it expresses the transactivator. And here we have a promoter, which is the 3G TET-inducible promoter, which then expresses the shRNA. Upon doxycycline, so this is a TET-on system here, then this transactivator binds to the TET operators in the inducible promoter and starts expressing these shRNA cassettes. What does it look like in real life? Okay, again, FACS profile. This is without doxycycline. This is a TAG GFP stable cell pool. It's not a cell line, it's a cell pool. And you see here, if you add doxycycline, you get a more of 100-fold increase in protein expression, and what is really nice is that the shape of the cell population stays homogeneous. The same is true for the TAG RFP, which is a little bit not so highly glowing or not so bright compared to the TAG GFP. Therefore, the induction is only approximately 20-fold in protein expression. But you experience very high degree of tightness and very high and homogeneous degree of inducible gene expression. That's what it looks like in a FACS profile. Again, no overlap between the parental cell population without doxycycline and the induced cell population with doxycycline. On the right, you see what it used to be before. Before, this is the second generation TET-inducible system, we had a very, let's say, 3 log units. This is more than 1,000-fold difference in gene expression within a cell population when you induced gene expression with the older systems. And I guess that's a very good improvement, and this allows us now to use those systems and to really do target research. The TET-inducible also works in the adenoviral vector. Here we have to use, basically it's the same expression cassettes, but we have to split them in 2 different vectors. This is nicely working, and as you may see here, if you add 50, 100 or 200 or 400 particles adenovirus per cell, and you induce the expression with doxycycline, you see that you nearly get a collinear increase in gene expression. This means that using adenovirus you have a further kind of application which becomes possible, so to get very low expression in a cell population to very high expression. So you can a little bit titrate the gene expression in the different cell types depending on the amount of vector applied. Okay, and again there is basically almost no background detected without doxycycline. More than 1,000-fold induction on the mRNA level of gene expression. Now coming to our target validation. So our starting point now is we have a technology which allows us really to generate stable cell models and inducible cell models. Now we need the RNAi, and now we need to be predictive at the end in the cell model. What is required? Okay, first we need to have a very good knockdown, more than 90%. Then we need to have the knockdown inducible. So I guess anyone that tried to have an inducible knockdown of more than 95% knows that this is a task. Then we need to get a real good cell model which has homogeneous cell expression. Okay, let's go started first with a kind of basics for RNAi experiments. We have provided, or we provide always some guidance to the customer. This means if you come with a target, we analyze the target and we check for the availability of good sequences. We tend to recommend to use more than one sequence. we will provide knockdown activity more than 90% on the target, and we can provide a verification on the phenotype by rescue and overexpression of the phenotype and the target gene respectively. And most of all, we can do it inducibly. So that's very important, because inducible gene expression in target validation has an advantage over constitutive approaches, because you can induce the phenotype, and then you can, by simply taking away the doxycycline, for example, you can revert the phenotype. So that's a kind of inbuilt control, and you a little bit also counteract adaptive cell responses, which sometimes occur when you have some cell mutants which can cope with the knockdown and then escape and show a phenotype which is kind of a little bit not representative for the gene knockdown. We use a standard control set. There are some, I will not go too much into detail here. Okay, how do we select very good sequences? So this is the core of the RNAI-1 technology. We start with bioinformatic algorithms which are very potent. You can have very potent ones from Whitehead Institute, Broad Institute. We take them all into consideration and generate a short list of approximately 100 pre-qualified shRNA sequences. Then we have a selection in-house, and we choose 10 to 15 designs of these pre-qualified shRNA sequences. What we then do, this is very important, we have a validation vector. We clone each shRNA into a single validation vector, and then we do in vitro analysis of activity. And this is what you see at the bottom. Here we tested 15 designs against a GPCR, which is notoriously difficult to knock down. And as you may see, even we used 15 top-ranked shRNA designs. Only 2 of them displayed in cell, so this means really under real conditions in vitro activity. Using these 2 sequences, we then generate viral vectors and then proceed with cell pool generation. So this is the result of one of these sequences. It is sequence number 15, and you see that you get a very nice inducible gene knockdown by addition of 0.5 microgram per milliliter doxycycline, and the remaining mRNA is only 10% of the wild type or non-knockdown level. And then we have done a comparison. We did, okay, what does it look like at the clonal level? So the pool shows 90% inducible gene knockdown, combination of our RNAI-1 technology, combination of our all-in-one inducible third generation TET-INDUCIBLE lentivirus vector platform. We analyzed 20 clones and analyzed the level of the inducible gene knockdown. And what you can appreciate here is that clone number 6, for example, shows as much as 98% of inducible gene knockdowns. So that's really outstanding, and we can use this clone now, for example, to generate screening or cell modeling or phenotypic analysis. But also the cell pool showing 90% of inducible knockdown is fine and yields very nicely, very nice results in terms of phenotypic. Here I throw a representation of 30 different gene knockdowns that we did using our RNAI-1 platform, and in green you see all the sequences which showed more than 90% of knockdown. So we have in this case a set of 10 shRNA sequences that we tested for 30 different genes, and the matrix already shows you that in some cases you only find one shRNA which has more than 90%, or in some cases like the gene number 8 you get a very high number of shRNA which knock down the gene by more than 90%. And now this is really the final result in the cell pool, meaning the product that you as a customer gets delivered. You get knockdowns which are either inducible or constitutive between 80 and more than 90%, and in more than 80% of the products delivered, the final knockdown in the cell pool is more than 90%. So what we do is we guarantee a knockdown of 80% in the final cell pool, but you may already see that you can expect more in your product. Now coming to the last part of our presentation, this is a VARICHECK. We use it as to really provide a kind of security in target research. This means we combine high on-target efficiency, we combine TET-inducible gene expression, and then in the same cell model we use inducible expression of a rescue variant. This means it's a protein variant that cannot be degraded by the shRNA expressed already in this cell pool or cell line. And what we should see at the end is a phenotype which is associated to the knockdown, and then the reversion of the same phenotype which is then induced by the expression of the non-degradable cDNA. Okay, let's go if it really works. So the workflow is the same. We start with RNAI-1, we do stable inducible knockdown cell line generation, and then in this intermediate knockdown cell line, which is already a very good first product, we simultaneously overexpress a protein, for example, that can be an RNAi on-target control, so meaning having some point mutations in the shRNA binding site so it will not be degraded by this shRNAs. Or what we can do, we can express protein variants like having distinct point mutations in order to analyze if these point mutations are associated, for example, with enhanced growth or resistance to a drug treatment. So that's a kind of screening, rapid screening model. And once you have identified here that, for example, one protein mutant is associated with resistance, then you can go on with genome editing and use the same cell model then really for drug discovery and screening. It's a schematic representation of what I explained. So without doxycycline, you basically only have the wild-type gene expressed. If you add doxycycline, you have the wild-type gene which is degraded by RNAi, and then you have an mRNA expressed which then is not degraded or which then represents a protein variant. Application for RNAi. This is our cancer target that we did in glioblast. No, it's in lung cancer cells. In this case, just let me confirm the basics. We start here. This is the starting material, plus doxycycline, minus doxycycline. This is the target gene. You have a very clear knockdown on protein level. So that's a very good starting cell line. If you knock down here the target gene with doxycycline, what you see is a decrease in cell proliferation. That's exactly the phenotype you want to see. Then we provide the control. In the same cell, we re-express the gene of interest, but having the point mutations in the shRNA binding sites, and then what we see is the re-expression of the protein, and this is really important, in blue, we get a reversion of the phenotype. And that means that the phenotype you see is really associated with the gene of interest or with the knockdown of the gene of interest. And this is what we call a real inbuilt control, which gives a high degree of confidence to your results. How does it work for you as a Horizon customer. So you would first of all transfer the information regarding your cell line of interest or your gene of interest to Horizon, and Horizon would transfer the same information to Thirion Biotech. We would do the feasibility check, then provided that the feasibility check is positive, so meaning that there are shRNA sequences, for example, which can be used, we would go into the selection of shRNAs, we would clone those shRNAs in the validation vector, perform the validation, do the quantification, and then we would just provide the lentiviruses or the cell models as wanted, provide a final report in the cell product, and ship the product back to you. We have maybe just it's always the best way to just is letting customers speak about you, and I would just give you some quotes from our Biotech, which is now Daiichi Sankyo. So here we support, for example, stable cell line generation and help them in target research. We are a partner. Then we have the company Brain, is a German white biotech company. Here we were involved in primary cell isolation and virus-based engineering technology, and we were able to help them to generate a very valuable primary cell model. And there are some academic customers where we provide adenoviral vector, also gave us a good reference. There are many more than, I have stopped counting, but more than 100 publications which demonstrate the value of our technologies. I hope this will be you when working with us. And now I would like to hand over to Chris, and I'm happy to take your questions.
Fantastic. Thank you for that presentation. I'm going to take control back, and I've just got a couple of slides that I'm going to go through very quickly around our offer. Both Thirion and Horizon are focused on the area of translational genomics, taking the information located in the human genome and through sequencing information and turning that into practical tools for drug discovery and development. It's often a very good first step when determining the function of a gene and whether or not a gene is involved in a disease or pathway to knock it down and look at the phenotypic effect. And shRNA knockdown is a perfect way to do that. The natural follow-on for that, if the answer is yes, and that it appears that a gene is important, is to fully knock it out or do further genomic modifications, and that's the part that Horizon provides. So Thirion and Horizon partnering together makes perfect sense from that perspective. Additionally, just the level of skill that Thirion brings to the table is incredible. The ability to knock down virtually any gene to a fidelity of over 90% is extremely powerful. On the right-hand side of the slide that you can see, there's a graphic which describes just how effective this can be. Taking a look at 30 different programs that they entered into, you can see that the vast majority of the time they're able to achieve an shRNA which has a knockdown of greater than 90% of the gene. So it leads to a very, very powerful set of tools which can provide great comfort and confidence when being used in an experimental program. What we offer in partnership with Thirion is off-the-shelf products and also a set of services. The off-the-shelf component consists of 125 shRNAs and around 75 overexpression products, which are available in two different formats, as lentiviral expression plasmids or as lentiviral particles, which are ready for transduction. And you can see on the slide what the prices are for those. Additionally, if the shRNA you're interested in is not available, the siRNA-1 program that they have can generate either a report, if you're just looking for information, then you can generate the shRNA yourself as an oligo form or any form you choose, or they will perform cloning and provide you with lentivirus, which is ready for transduction. Either the best performing, the 3 best performing, or the 5 best performing, depending on if you wanted to do pools or provide backup for your experiments. And they can go a little bit further and generate cell pools as well. Now, if those cell lines that your cell pools are going to be generated in are ones that they're comfortable with and they have deep experience with, there's no additional charge. But there is a small charge associated with new novel cell lines, ones they haven't worked with as much, so that they can confirm that the transduction is going to work and that they're able to perform their experiments to the level that is expected.